Welcome. Thank you all for joining us tonight. We're really excited you guys could come see this graduate student talk here in our galleries. And we're also really excited to welcome our speaker tonight, Irene Zhang. She is a PhD candidate in the microbiology department here at MIT, or microbiology program here at MIT. Irene also works with the Earth, Atmospheric, and Planetary Sciences department at MIT, and her research really concerns the metabolic interactions between microbes. So tonight we'll get a chance to hear a bit more about Irene's research as it relates and overlaps with some ideas explored here in our exhibit on view, Symbionts, Contemporary Artists in the Biosphere. And Irene will specifically be examining the work of Anicka Yi. This work is called Living and Dying in the Bacteriacene. So through the lens of her research, we'll learn a little bit more about this piece as well. So without further ado, let me hand it to Irene.
Thank you. Hi everyone, thank you so much for coming here. I really appreciate it, especially in this weather. It's not the greatest, but I really appreciate you coming here. And also, I would like to thank the List Visual Arts Center for putting on this amazing exhibition. So if you have a chance afterwards, I would really encourage you to take a look around. And also, I would particularly like to thank Elizabeth Ponce, who was just talking. She is the public programs coordinator. Correct me if that's not right. But she was responsible for reaching out to me and getting everything set up for this graduate lecture series. So I really appreciate that as well. And yeah, so as you know, the theme of this exhibit is called Symbionts, Contemporary Artists in the Biosphere. And so all of the work in this exhibition in these galleries relates back to that central idea of symbiosis. And in particular, also this piece here by the Korean-American artist Anicka Yi. And so I was really interested in this piece and attracted to it like when I was first looking through the exhibition catalog, because I've actually been following Anicka Yi's work for a long time. I actually follow her Instagram because I've been interested for a long time in this idea that all of her art sort of draws on these concepts from biology and from chemistry, which is kind of close to me because I majored in both biology and visual arts as an undergrad. So I'm actually really excited to talk about this because it's not very common during the course of my PhD for me to kind of like merge these two interests of mine together. So this is actually like super cool opportunity. So thank you. And so this piece is called Living and Dying in the Bacteriacene. And so within this piece, it's sort of like an aquarium in which there is water, and then there's also this living filamentous algae that you can see growing, and it's actively photosynthesizing within this piece here. So that's really interesting to me because marine algae and photosynthetic phytoplankton actually are responsible for a huge portion of oxygen on Earth, 50 to 80%, which is more than actually all terrestrial plants and trees combined. And they're also responsible for half of global primary productivity, which is the fixation of carbon dioxide into biomass, which is organic matter, everything living. So they're actually hugely relevant to the biosphere and kind of underappreciated when people talk about like carbon and productivity and oxygen. A lot of us think about trees, but these organisms are actually really important for those processes that sustain life. But when you look at this, so the media that Anicka Yi says the only media that she kind of talks about as living are these algae. But actually these algae aren't the only things that are growing within this artwork and within this environment. So for example, if you took a small sample of water or of the algae itself and you did DNA sequencing, you would find that there's like this huge associated complex microbial community that consists of hundreds or thousands of different taxa of non-photosynthetic bacteria and eukaryotes, as well as archaea, fungi, and viruses. And this is actually true for like most microbes that live in the environment or anywhere. They usually live in these communities that are really complex, and there's a lot of them out there. So for example, if you went out into the water, like the seawater, and you took like just a couple of drops, like 1 milliliter, and you looked at that under a microscope, you would actually find that within just that amount of water, there's around 1 million bacteria, and on the order of like 10 million viruses. So yeah, just think about that the next time you go swimming in the ocean and you're like ingesting water. So yeah, it's really cool what these organisms are doing in the environment. And like one of the major things that I've been focusing on during my research is just figuring out who is there and like what are they doing. So within this algae and within like all major, the different species and groups of marine algae and phytoplankton, there's actually this really consistent ratio of the major elements. So carbon, nitrogen, and phosphorus are like the three major elements that make up all life. And within marine algae and phytoplankton, the ratio of these elements is kind of very similar across a lot of different organisms. It's this ratio that's called the Redfield ratio, and it's 106 carbon atoms to 16 nitrogen atoms to 1 phosphorus atom. And so all the elements of this ratio, which are like the same elements that make up like you or me, these elements are actually being cycled and being fixed and being converted between their different forms, mostly by microbes, or a lot of it at least by microbes. So as I mentioned, the carbon in this is being fixed from carbon dioxide, which is like present in the atmosphere, by these organisms such as like photosynthetic microbes, but also the nitrogen is actually being fixed primarily by microbes as well. So nitrogen, as you know, makes up like 70% of our atmosphere, so it's like a huge part. But actually the primary form of nitrogen in the atmosphere, which is N2 gas, dinitrogen gas, is biologically inert. So organisms can't really use it, they can't really metabolize it, it's just kind of there, but not usable. So in order to turn this nitrogen into forms that people and other life can actually use, such as amino acids that make up like proteins, and also nitrate, which is like a major nutrient. It's like a really important part of fertilizer and agriculture. You have to have these specialized organisms called nitrogen fixers. And so you can kind of think of this process as really similar to the fixation of carbon dioxide, but instead they're fixing nitrogen. And this is actually like a really specialized process because when you think about like the two atoms of nitrogen gas, they're connected by this really strong, like really tight triple bond, which requires specialized enzymes to break. And a lot of these enzymes are carried by microbes in the water, in the environment, et cetera. And then so they cleave this bond and they turn this nitrogen into forms that other organisms can use, such as you or me. And this is like true for a lot of the different elemental cycles on Earth. Microbes do a ton of it. So, however, if you think about this process just like constantly happening, like this nitrogen from the atmosphere is being converted into these forms that we can use, and then we're eating it, we're doing all these things with it. If you don't have like kind of the opposite process going on, then you would find that the 70% of nitrogen that's in our atmosphere would start decreasing, which is not very good for our climate. So that's kind of the part of the nitrogen cycle that I personally study. It's this process called dehydrification, in which these bioavailable forms, such as nitrate, are then converted back into nitrogen gas and then emitted into the atmosphere. And this is the way that the nitrogen budget is kind of like balanced between the losses and gains of each of these different forms. And this is a process that's entirely microbial. It's carried out by these facultative anaerobic microbes that are kind of like breathing nitrate instead of breathing oxygen. So similar to like how you or I would like breathe oxygen and get energy from that, these organisms, when the oxygen is really low, they can switch from breathing oxygen to breathing nitrate. So this process kind of happens in areas where oxygen is kind of low. And if you've heard about like marine dead zones or like ocean dead zones, which are like areas of the ocean where the oxygen is so low that normal aerobic life forms like fish can't live, these are actually not really dead if you look at the microbial community, because there's actually this like very abundant, very diverse, and like very active microbial community in those areas in particular. And all of them are kind of cycling these things like nitrogen, sulfur, phosphorus, et cetera. So these areas of the ocean like are actually kind of really important for maintaining our atmospheric nitrogen balance and also just for cycling these nutrients in general. So that's like kind of what I study, because I study dehydrification in these ocean so-called dead zones or oxygen deficient zones. And historically, it's been hard to kind of study them because they like tend to be kind of offshore and several hundred meters below the sea surface. So it's hard to sample them. And also the microbes that are living there are hard to culture in the lab because they tend to be like slow growing. They need special nutrients or special media in order to grow. And so it's just like really hard to kind of study them without DNA sequencing. But the recent advances in DNA sequencing have allowed us to take DNA from this area and then kind of reconstruct the genomes of the organisms that are living there. And then based on the genes that they have, we can kind of tell like who's doing what process, where they're doing it, how much are they doing it, et cetera. So we can kind of scale up from the lowest, the like smallest scale of the single cell all the way up to like the climate level, the global scale of the cycling of different forms of nitrogen and carbon, et cetera. So like through my research in the Earth, Atmospheric and Planetary Sciences department, I'm trying to kind of connect these two scales that go from like the very smallest form of life on Earth all the way up to the Earth itself. However, you might like think that this like happens like super far away and like doesn't really have any relevance to us. But the same microbes, or like not exactly the same ones, but the ones that are doing these processes are also present in each of us here. So the human body is actually mostly microbial because there's more microbial cells than human cells in the human body. And so these microbes, such as in your gut, are interacting with each other and with you through your immune system, through like what you eat, et cetera. And they're kind of all living and cooperating and coexisting and co-evolving together. So this is kind of like this idea of like the holobiont, which is this like set of interacting organisms that are all co-evolving like with each other. And you can't really like take any individual organism from this group and look at it like just in isolation, like by itself, without any of its like interacting partners. So like when you think about this community level idea of like evolution and like agency, you have to sort of update this Darwinian like survival of the fittest concept in which each organism is like kind of out for itself. Like it's trying to like outcompete everyone else, it's trying to be the fittest. But actually like when you look at the community, it's not about like which individual organism is like the fittest, but it's more about like which community is best adapted to like whatever environment it's living in. And there's like a lot of ways that you can kind of think about this. So like what is the best adapted community? Like is it the most diverse one or the one that like cooperates with each other the most? Where all organisms sort of like working together towards this common goal of like survival? Or maybe it's like the one that's like resilient against like stresses, or if the environmental conditions like fluctuate, that's the one that's going to survive, whereas other communities that might not be as resilient might go extinct and die out. But it can also just be like pure luck, like whoever gets there first in whatever environment then can take maximum use of whatever nutrients are available and prevent other invaders from coming into that niche and like kind of proliferating within it. And sometimes it's just like pure luck, like whatever is there at the right place, the right time is what survives. So we don't really know a lot about these processes because they've been like historically pretty hard to study in the lab, but our knowledge of this is like growing every day as people use like sequencing technologies and other like approaches to kind of study how these communities like have these ecological processes that enable them to coexist and co-evolve together. So with that, I just like to bring us back to this work here, which is called Living and Dying in the Bacteriacene. I think I mentioned that before. But there's a couple ways that I like to like interpret this, especially like the title, because I think the title is really interesting. So there are a couple of ways, and like you can kind of agree or disagree or like come up with your own ways. But basically what I think about is that when you look at this process, like when you look at this ecosystem, and this is like sort of like a small self-contained ecosystem by itself, like within the bounds of the piece of art itself. And you can kind of think of like the organisms in here as like part of a holobiont because they're all interacting with each other and living together in this community. And so we might like look at it and think that it doesn't really change like by like day by day or like hour by hour, but actually there's like a really dynamic process that's going on in here in which the microbes that are present are cooperating with each other and exchanging nutrients, metabolites, DNA, protein, whatever. They're also competing with each other for different resources because the nutrients in here are limited and they have to kind of compete for who gets those nutrients. And the ones that don't win this competition might go extinct or die out. So this holobiont is also like dynamic. It's not the same, like no matter what time you sample it. Then there's also like more active ways that these microbes are killing each other, just beyond just like competing, because microbes actually like are the source of a lot of our antibiotics, like penicillin, and these are chemicals that microbes themselves synthesize in order to like kill other microbes that are around them in their environment that might compete with them. And then there's also other ways, such as like some microbes have evolved this system in which they actually shoot out this like needle and they can puncture other microbes similar to like how you might puncture like a water balloon. Then other microbes might be eating each other and engulfing each other and digesting those like whatever they eat as sources of nutrition. And also within this, there are microbes that are actively replicating and procreating and forming new generations of microbes. So this is actually a really dynamic process, and the microbes within it are like living and dying as we speak. And then, so another way that I think about this is through this concept of the Bacteriacene. So you might have heard about like the Anthropocene, because that's like what people call the current age we live in, because like humans have caused so many changes in our climate, in the ecosystems, and actually we're causing the ongoing sixth mass extinction due to things like anthropogenic climate change. But if we rewind time back 2 billion years ago, there was actually an even deadlier mass extinction event that was caused entirely by microbes, mostly bacteria. And so prior to that, the Earth's atmosphere actually contained very little oxygen. It was primarily anaerobic. So the microbes that were living there were doing these anaerobic processes, such as like nitrogen, sulfur, phosphorus, like hydrogen, like using those things instead of oxygen for living. But then there were some microbes that evolved this ability to do photosynthesis, basically. And the result of photosynthesis is oxygen. So they were accumulating this oxygen in the environment and changing the entire atmosphere of the Earth from one that had very little oxygen to one that had like a lot more oxygen. And this is like what people call like the great oxygenation event. But also, oxygen is really toxic to microbes that aren't aerobic. Like it's a really toxic compound. It kills like their metabolism and poisons their enzymes. So this actual like accumulation of oxygen in the atmosphere caused the anaerobic microbes that were present to like mostly die out actually. Like whole lineages went extinct, whole families went extinct. And so this was why it's like a mass extinction event. But what this caused was the ability for the proliferation for a lot of aerobic life forms, such as us here, and plants, animals, like insects, whatever, all of these other things that breathe oxygen. So instead of thinking about like the Anthropocene, maybe we can think about basically every single epoch on Earth as the Bacteriacene. Then finally, the last thing I want to talk about is the fact that this isn't just about like something that happened 2 billion years ago. This is also about something that's happening right now in each of us, because the human microbiome obviously is this like super hot topic right now. And we are all holobionts, we're all ecosystems, because within our guts, we have this gut microbiome that helps us digest like all these different foods that we eat. We also have like skin microbiomes that protect us from invading pathogens. We have like sinus and lung microbiomes that help prevent us from developing these diseases such as asthma and sinusitis. And there's even this idea of the gut-brain axis, in which our gut microbiomes affect our brain development, our moods, and also might impact our ability to get like Alzheimer's or other diseases later in life. So microbes aren't just like pathogens or germs that we should get rid of. And if we actually did get rid of them, like if you just removed every microbe from your body right now, you might actually die, or at least you would live like a very different life from what you're doing right now. So they're beneficial, but there's a fine line between like what is beneficial and what is like a disease or a pathogen, because the same microbes that are within you, if given the opportunity, might grow out of proportion and then cause all of these diseases. Because our diseases are also mostly microbial. Like we have these fungal infections, like these bacterial infections, viral infections, COVID, et cetera. So they're both harmful and helpful to us. And they are really tightly interwoven with our process of like living and dying. Like if you died right now, the same microbes that are within you would start digesting your body. So yeah, I guess it's interesting to think about the fact that we're like living and dying in such close connection with these symbionts that are within us, on us, and all around us. And so with that like final thought, I just kind of want to bring us back to this idea of like the Symbionts exhibit, because it lets us kind of think about all of the ways in which we interact with these organisms that are all around us. So for example, like the organisms that we can see, such as in the spiders across the hall, like all the way over there, those are symbionts within our homes as well. But there's also a lot of invisible symbionts, such as these microbes, that maybe you can like think about as you're going about your daily lives. And I also want to encourage you to look at some of the other work in this exhibit, such as the soil erg pieces by Claire Pentecost. They're like in that room over there, as well as there's an exhibit called Plantas Autophotosyntheticas by Gilberto Esparza. That's like in a room like over there, I think. And these are also works of art that talk about the ways that our structures and our society are so tightly interwoven with the microbial symbionts that inhabit this earth. And yeah, if anyone has any questions, I would be happy to answer them at this point.